In this video, we're going to go through meiosis, which is one of the more challenging topics for paper one. So we're going to go through the stages of meiosis, what happens in meiosis 1, meiosis 2, focusing on crossing over and independent segregation or independent assortment. So stay tuned if you need help with this topic. First of all, why is meiosis important? Well, we should already know from GCSE that meiosis produces 4 genetically different gametes or sex cells. So obviously we're thinking of sperm cells and egg cells. They will all be genetically different. So offspring produced will be genetically unique. They are haploid cells, which means they have a single set of chromosomes represented by N. Remember, mitosis produces diploid cells, which we represent with 2N, because in mitosis, the cells produced have pairs of chromosomes. The chromosomes are still in homologous pairs. And mitosis also makes genetically identical cells, whereas meiosis, as we've said, makes genetically different cells. Now, why is it important that gametes are haploid? Well, obviously, gametes are used in fertilization. So fertilization is when a sperm cell, the male gamete, fuses with the egg cell, the female gamete. And when they fuse at fertilization, because they were haploid, so in humans, they each had 23 single chromosomes, that means that the zygote, or the fertilized egg cell, will have the correct number of chromosomes. So we can say that using haploid gametes will maintain the chromosome number at fertilization. It's really important that you say at fertilization, these gametes are going to fuse. And because the gametes were haploid, this will maintain the correct number of chromosomes in the zygote. Obviously, once we've made the zygote will then divide by mitosis, not meiosis, mitosis to grow and develop into the embryo. So all the cells of the embryo will then be genetically identical and will be diploid cells with 46 chromosomes. So meiosis only occurs in the testes of the male, the ovaries of the female, just to make the gametes. So the gametes are haploid and genetically unique, genetically different. Let's go through the stages of meiosis. Now, one of the differences between meiosis and mitosis is that meiosis consists of 2 divisions, and this is why we end up with 4 daughter cells or 4 gametes produced from 1 body cell. And it's meiosis 1 that is really important to understand, because it's meiosis 1 that differs from mitosis. So let's go through what happens during meiosis 1. And just to mention that similar or same as mitosis, it is preceded by interphase. So both mitosis and meiosis are preceded by interphase. Interphase is obviously when the chromosomes are replicated or the DNA is replicated. The cell also increases in size as the cytoplasm increases in volume. Ribosomes are replicated, mitochondria are replicated. That's interphase, and that is common to both mitosis and meiosis. It happens before both of them, right? But what happens during meiosis 1? So let's go through. So we still have the same phases, if you like. We have prophase, metaphase, anaphase, and telophase. Prophase 1, the first prophase, meiosis 1 prophase, this is different, right? Because homologous pairs associate. They come together and form what we call bivalence. This does not happen in mitosis, okay? In mitosis, the homologous pairs do not associate. They do not form bivalence. And you can see a bivalent here. So this is a bivalent, this is another bivalent here. It's just the homologous pairs actually come together in their pairs, they associate together. And this is when crossing over can occur. Not always, it's infrequent, but crossing over can occur. And we'll talk more about that later in this video. So that's prophase 1. Obviously, same things as mitosis in terms of chromosomes condense and become visible, nuclear envelope breaks down, but we already know that from mitosis. So we're really just focusing on the differences between mitosis and meiosis. Yes, the nuclear envelope breaks down. Yes, the chromosomes condense and become visible. But in meiosis, in prophase 1, the homologous pairs associate and form bivalents, and that's when crossing over can occur. Now, during metaphase 1, we know metaphase begins with an M, middle begins with an M. We know this is about them lining up on the equator, but what is different? In meiosis 1, the bivalents line up at the equator, or the homologous pairs of chromosomes line up at the equator. They line up in their pairs. This does not happen in mitosis. In mitosis, they all line up individually, but you can see on the diagram, meiosis 1, metaphase 1, the homologous pairs are lining up at the equator, and this is when independent segregation, or you might call it independent assortment, takes place, which we'll cover later. Now, obviously, similar to mitosis, they are attached to the spindle fibers via their centromeres, but the difference is they're lining up in their homologous pairs and they line up randomly, which is independent segregation. Then we have anaphase 1, and we can all recognize anaphase. We know this is where chromosomes are being pulled to opposite poles, right? But with mitosis, the centromeres split and it's the sister chromatids that are pulled to opposite poles. In meiosis 1, it's the pairs, the homologous pairs of chromosomes that are separated and pulled to opposite poles. Again, it uses the spindle fibers, but if you have a look at the result, we have separated the pairs of chromosomes. So the centromeres have not split in anaphase 1. The centromeres are still intact and each chromosome still consists of 2 sister chromatids held by that centromere. We have not separated the sister chromatids, we have just separated the homologous pairs. So one from each pair will be pulled to an opposite pole. So that's anaphase 1. Then we get to telophase 1, which is kind of similar. We've got the nuclear envelopes reforming. So at the end of meiosis 1, we'll have 2 new nuclei. Nuclear envelopes reform and the chromosomes decondense. So you won't be able to see them again for a short time before meiosis 2. But yeah, we've now ended meiosis 1. We've got 2 new nuclei. Not 4 yet because we need to go into meiosis 2. We've got 2 new nuclei, but the key to meiosis 1 is we have separated homologous pairs. So the cells are already haploid. They only contain 1 chromosome from each homologous pair. So by the end of meiosis 1, we've got 2 nuclei. They're already haploid. We've separated homologous pairs from their partnerships, yeah, because the bivalents lined up together. Right, then we can move on to meiosis 2 because there's a second division. But luckily for you, meiosis 2 is just like mitosis. You don't really have to learn this. You don't really have to learn the stages. You just need to know that there's a second division. But this is now just like mitosis, except we no longer have homologous pairs of chromosomes. So prophase 2, same as mitosis, chromosomes condense and thicken, just like they do in mitosis, just like they did in meiosis 1. But we've already separated the pairs, right? So in metaphase 2, it's the chromosomes that line up on the equator, obviously attached to spindle fibers via their centromeres. And then anaphase 2 is like mitosis. The centromeres now split and the sister chromatids are pulled to opposite poles via the spindle fibers. And then telophase, nuclear envelopes reform. Obviously, the chromosomes will decondense and will not be visible, but now you've got 4 nuclei. Now, hopefully what you've noticed by doing this with me is meiosis 1 is the key. Meiosis 1 is different to mitosis. Meiosis 1, let's just go back for a second. Meiosis 1, the homologous pairs associate and form bivalents. We get crossing over. The homologous pairs line up in their bivalence at the equator. We get independent segregation. Then we only separate the pairs of chromosomes. We do not separate the sister chromatids at this point. Then, moving into meiosis 2, just like mitosis really, this is where now the chromosomes, the individual chromosomes, line up at the equator, the centromere is split, and the sister chromatids are pulled to opposite poles. Now, because we have 2 rounds of division, we end up with 4 daughter cells, not 2, like we do in mitosis. All the daughter cells will be genetically unique because we've had crossing over and we've had independent assortment, and they will be haploid. They will consist of single chromosomes because of what happened in meiosis 1, where we separated the homologous pairs. The chromosomes are no longer in their homologous pairs. Now let's add on a little bit, because the questions do often focus on why meiosis gives rise to genetic variation. Why are the gametes genetically different? So let's talk about that. The first thing you're going to name, if you're asked to state a reason, is crossing over. Crossing over can occur, and this involves recombination of non-sister chromatids. I'll show you that on the diagram in a second. It occurs during prophase 1, so meiosis 1, prophase 1. And the end result, which is really important that you learn this phrase, is you get new combinations of maternal and paternal alleles. Let me try and show you. So here you go. You've got a pair of chromosomes. This is a homologous pair of chromosomes. And all that means is they carry the same genes in the same loci. But remember, they could have different versions of those genes. They could have different alleles. Now, the reason the homologous pair looks like this is obviously after DNA replication. So we've already had interphase, we've already replicated the DNA, and that's why each chromosome consists of 2 sister chromatids held together by the centromere. Okay? So this is 1 chromosome in the homologous pair. This is the 2nd chromosome in the homologous pair, but each chromosome consists of 2 genetically identical sister chromatids because it's after DNA replication, and they are held together by the centromere in the middle. Now, crossing over occurs, remember, during prophase 1. So during prophase 1, we said that the homologous pairs associate or form a bivalent. This is a bivalent, yeah? The pair of chromosomes has come together. And when that has happened during prophase 1, you can see that you've got a crossover point here. This is called a chiasma. Yeah, you might have multiple cross points, chiasmata, but here we can just see 1 cross point here. So 1 singular is a chiasma. Now, when a chiasma forms, we can say portions of chromatid break off and reattach to a different chromatid, if that makes sense. Now, I just want to talk to you about what non-sister chromatid means here, and hopefully you'll understand this. These are sister chromatids, right? They are genetically identical because they're the result of DNA replication. These are also sister chromatids, right? They're genetically identical as a result of DNA replication. What we say for crossing over is that portions of chromatid are exchanged between non-sister chromatids. So you've got a portion of the blue chromatid breaking off and a portion of the purple chromatid breaking off. And if you look here, the portion of purple chromatid has attached to the blue chromatid. That was a non-sister, non-genetically identical chromatid. It may have had different alleles, and the portion of blue chromatid has attached to the purple chromatid. So non-sister chromatids, that's where it occurs. So portions of chromatid break off and they can reattach to a different chromatid. I'll just add on, it will be a non-sister chromatid. And now these chromatids have new combinations of alleles. I wonder if I can actually show you this by putting some alleles onto here. I'm going to give it a go. So let's imagine there's a dominant A allele here. This would also be a dominant A allele because it's genetically identical. They are sister chromatids. Let's imagine there's a recessive, oh no, let's make it dominant. A dominant B allele here and a dominant B allele here. Now this is a homologous pair, so this will have the same genes in the same loci, but they could be different alleles. So let's imagine we've got 2 recessive A alleles for that gene there and 2 recessive B alleles for that gene there. Same genes, same location, but in this homologous pair, we've got different alleles, different versions of those genes. Let's put them onto here in the same positions. And let's show the result of crossing over. So here we've still got the dominant A attached with a dominant B, but here we've got the dominant A that was on the purple chromatid and now it's attached to a recessive B. Here we've got a recessive A. This one is still attached to a recessive B, but here, because portions of chromatid have been exchanged, this recessive A is attached to a dominant B. So now you've got different combinations of alleles. You've got this new combination and you've got this new combination that if you have a look at the chromatids prior to crossing over, you did not have those 2 combinations. You only had these 2 combinations, okay? Let's just recap key points here. It's called crossing over. It introduces genetic variation into the gametes. It's prophase of meiosis 1. We can say chiasma form, portions of chromatid break off, and you can just say and are exchanged if you want to. That's a nice simple way to phrase it. You get chiasma forming or chiasmata forming, portions of chromatid break off and are exchanged between chromatids, non-sister chromatids if we're being really picky, but you don't really have to remember that for AQA. And the end result is new combinations of alleles. So important that we say new combinations of alleles. And we've shown that here by adding an example. You can see the new combinations of alleles. You've now got a dominant A with a recessive B and a recessive A with a dominant B. You did not have those combinations of alleles before. So this gives rise to genetic variation. Okay, let's see if we can explain independent segregation as another way that meiosis introduces genetic variation. Now, independent segregation, independent assortment, takes place during metaphase. Again, it's meiosis 1. Metaphase of meiosis 1. I mean, you can also say in meiosis 2, because I suppose the sister chromatids could be separated into either daughter cell, but we're going to focus on meiosis 1 because that's where it comes up in the exam. Remember, during metaphase of meiosis 1, it's homologous pairs of chromosomes that line up along the equator and they line up at random. So let's have a look at it on the diagram with just 2 pairs of homologous chromosomes. So if they line up like this, you've got the purple chromosome on the left and the blue on the right for that pair. You've got the red on the left and the pink on the right for that pair. But they could line up like this. So the purple's on the left, the blue's on the right still, but look, the pink and the red have lined up in a different order, a different orientation, and that's all it is. So when during anaphase 1, we obviously separate the pairs of chromosomes. If they had lined up on the equator in this orientation, these would be the 2 daughter cells, right? So you can kind of picture it like this. You'd have the purple with the red and the blue with the pink. But if they lined up like this, you'd have these 2 daughter cells. You'd have the purple with the pink and the blue with the red. Now, this doesn't look like a lot of genetic variation, right? But you've got to remember there are 23 pairs of chromosomes lining up on the equator during metaphase 1 of meiosis 1. And in each of those pairs, they can line up in either order. There's two options, if you like, for each of those 23 pairs. Now, this is called independent assortment or independent segregation. And what you need to say is that the homologous pairs of chromosomes line up in their pairs at random along the equator, so that when they are separated, we get different combinations of maternal and paternal chromosomes. Again, this as an explanation for how it gives rise to genetic variation is so important. So I used colours to explain it more simply. But the purple chromosome here originally came from the father, the blue originally came from the mother. That's obviously what paternal and maternal means. Now, if they lined up like this, you can see that the two paternal chromosomes go into a daughter cell and the two maternal chromosomes go into a daughter cell. But if they line up like this, you've got a paternal and a maternal in this daughter cell and a maternal and a paternal in this daughter cell. And remember, you've got 23 pairs of chromosomes lining up. There's two different ways they can line up. So you've got different combinations of maternal and paternal chromosomes. That's what you need to be able to say. Okay, so crossing over, prophase 1, independent segregation, metaphase 1, meiosis 1, new combinations of alleles for crossing over, new combinations of maternal and paternal chromosomes for independent segregation. Both give rise to genetic variation in meiosis. Now, one more thing before we finish, there's some maths that we can do linked to independent segregation. So let's see if you've done this before. It is possible to calculate the number of possible chromosome combinations that can arise from independent segregation. Now, the formula to do this is 2 to the power of n, where n is the number of pairs of homologous chromosomes in the original body cell, in the diploid cell, in the ovaries, in the testes, in that cell that is being used to make gametes. So we can calculate the number of possible chromosomes in a human egg cell by doing 2 to the power of 23, because in the cell, in the ovary, that is undergoing meiosis to make the egg cells, there are 23 pairs of chromosomes. So 2 to the power of 23 gives us possible chromosome combinations of this many. Huge number of different combinations, because there are 23 pairs and there are 2 different ways that each of those pairs can line up on the equator during metaphase 1. Now, from this formula, we can calculate the number of possible chromosome combinations produced from random fertilization of gametes. Now, to do this, we do 2 to the power of n times 2 to the power of n, remembering n is the number of pairs of homologous chromosomes in the diploid body cell that is making the gametes. So in a human zygote, which is a fertilized egg cell, so the first cell that forms after fertilization, the egg cell fusing with the sperm cell. So there are 2 to the power of 23 different possible chromosome combinations in the egg cells, and so we multiply, there are 2 to the power of 23 different chromosome combination possibilities in the sperm cell. So that gives us, let's just leave it there at 3 decimal places, this many different chromosome combinations in the zygote. I'm just checking that I've done that correctly because I was speaking and calculating at the same time. Yeah, that many different combinations. And this is just the result of independent segregation. This does not even take crossing over into account. This is just the number of different combinations of maternal and paternal chromosomes that can be present in the fertilized egg cell or the zygote. Obviously, this is for humans, but you've also got crossing over to factor into that. So huge amounts of genetic variation. Let's just do a question here to finish. Calculate, shrink my face down so you can see the full question. Calculate the number of possible chromosome combinations from the random fertilization of 2 gametes that each have 5 chromosomes. So the gametes have 5 chromosomes. That means that in the body cell that made those gametes, there must have been 5 homologous pairs of chromosomes. The gametes just have 5 single chromosomes, but there would have been 5 pairs of chromosomes in the diploid cell that made them. And we're calculating the number of combinations from random fertilization of 2 gametes. So 1 gamete, combinations would be 2 to the power of 5, and it's going to fuse with another gamete that had 2 to the power of 5 possible combinations. So let's multiply that together, and we get a total number of possible chromosome combinations of 1024 in that zygote. Remembering that does not take crossing over into account, so actually there's going to be more genetic variation than that. Hopefully you found this video useful. Let me know in the comments if it's helped you to understand meiosis, independent segregation, crossing over, and the maths. And let me know if there's any other videos that you want me to post to help you with your revision.